Extensions of Mendelian principles, incomplete dominance and co-dominance in early 1090s, 1900s, when German botanist Karl Corens would repeat Mendelian crosses with a 4 o'clock plant, Mirabilis jalapa. He found a blending inheritance, which is exceptions to Mendel finding. The Korans introduced a term called incomplete dominance, which suggests the characteristic expression expressed in the F1 generation is neither of the parent type but a mixture of both. For example, when a red flower 4 o'clock plant RR is capital R is crossed with a white colored plant smaller, all the plant appears in the F1 or having pink capital smaller flowers. When the F1 plants are self-crossed, it yields red capital and pink capital smaller and white smaller flowered plants in 1:2:1 ratio in F2 generation. This is observed as a capital alleles is incompletely dominant over small or alleles. Now we know capital alleles encodes for pigmentation factors, whereas smaller alleles does not code for any pigmentation factor. In case of homozygous dominant capital, the pigmentation factors are produced in double amount, while in case of homozygous recessive smaller it is not produced. Thus homozygous dominant capital or red and recessive and or white without pigmentation. Interestingly, in heterozygous capital smaller, half of the pigmentation factor are produced, as there is only one capital or others, and thus they becomes pink. There is another explanation of Mendelian inheritance known as a codominance. When in F1 hybrid both the alleles are expressed, this is called codominance. It is observed in human blood group system as well as coat color inheritance in cattle. For example, when the red hair cattle capital is crossed with a white hair cattle smaller, the F1 progenies becomes roan color capital smaller. When these are self crossed, it yields red capital and roan capital smaller and white smaller colored cattle in 1:2:1 ratio. Here the alleles for red hair color capital or and alleles for white hair color smaller or expressed equally, and thus all heterozygous individuals have roan color capital smaller back cross and test cross. When a cross is set up between F1 hybrid and any of the parent, this is called as a back cross. On the other hand, a cross between F1 hybrid and homozygous recessive parents is known as test cross. Thus, we can say the entire test cross or back cross, but not the vice versa. For example, a cross between the hybrid tall pea plant capital T small t and either pure tall capital T or dwarf pure dwarf small t is known as a back cross. While if the cross is specifically happening with the homozygous recessive small t, it is called as a test cross. The test cross is very much important for determining the unknown genotype. According to Mendel's laws of dominance, we have seen only dominant alleles show their phenotypic expressions at F1 generations. We don't have any idea about its genotype. For example, both capital T and capital T small t are tall pea plants, and to know exactly whether the F1 progeny is capital T or capital T small t, we need to do test cross. If the monohybrid cross between F1 progeny with pure dwarf plant small t give rise to all the tall plants, it indicates the genotype F1 progeny is capital T, while if the cross produces tall and dwarf plants in 1:1 ratio, it indicates the genotype of the F1 progeny is capital T small t. It is in case of dihybrid cross, the ratio will be 1:1:1. Gene interactions. The single gene may interact with various ways, such as out of two alleles of single gene, one allele shows a simple dominance over the action of other, which is recessive, or both the alleles must have incomplete dominance and may have co-dominance relationship. Such kind of genetic interactions that occurs between the two alleles of a single gene are often referred to as interallelic or introgenic interactions. In case of intergenic interactions or non-allelic interactions, the independent gene may be located on the same or different chromosomes and interact with one another for the expression of single phenotypic trait of organisms. A gene or loci which suppress or mask the actions of gene at another locus is termed as a epistatic gene. On the other hand, the gene locus which is suppressed by an epistatic gene is called as a hypostatic gene. In case of dihybrid cross, when the epistatic interactions occurs between the two genes, less than four phenotypes may occur in the F2 generations. Such biogenic epistatic interactions can be explained by the following interactions. 1. Dominant epistatic phenotypic ratio 12 is to 3 is to 1. Capital A small a, capital B small b crossed with capital A small a and capital B small b. It forms capital A, capital A, capital B, capital B. And in case of recessive, capital A, capital A, capital B, small b, capital A, small a, capital B, and capital A, small a, capital B, small b. And next, say AB, capital A, small b, capital A, capital B, small b. The two recessive epistasis, the phenotypic ratio 9 is to 3 is to 4, and a duplicate recessive epistasis, the phenotypic ratio 9 is to 1. Interaction, the homozygous recessive of either gene capital A or capital B produces white. 9 is to 7, 9 purple and 7 white. And 4 duplicate dominant epistasis, 15 is to 1, 15 triangular and 1 ovoid, and 5 duplicate genes with a cumulative effect phenotypic ratio 9 is to 6 is to 1. 6 dominant and recessive gene interactions, 13 is to phenotypic ratio 13 is to 3, 13 white and 3 colored. Pleiotropism or pleiotropy, the phenomenon by which a single gene can regulate the expression of multiple phenotypic trait, it is known as a pleiotropism. For example, in case of human brain, a reaction that converts the phenylalanine to tyrosine is very crucial for behavioral and pathological perspective. This reaction is catalyzed by an enzyme, phenylalanine hydroxylase. The mutation in the gene encoding the phenylalanine hydroxylase leads to the deposition of phenylalanine in the brain. Because of this, a person starts developing abnormally and his hair and skin color changes. This disease is known as the phenylketonuria, and here the gene encoding the phenylalanine hydroxylase acts as a pleiotropic gene. In case of hemoglobin, the beta globin gene also acts as a pleiotropic gene. In sickle cell anemia, a missense mutations in beta globin gene leads to multiple physiological problems such as liver failure, eye damage, and heart attack. Genomic imprinting. The genomic imprinting is an epigenetic phenomenon in which the genes are expressed in a parent of origin specific manner. In case of mammals, the function of genomic imprinting is not well understood, but it may help a vital role in development. The recent research suggests that it is also responsible for uniparental inheritance. The most important disease caused by the genomic imprinting in humans are Angelman syndrome and Prader-Willi syndrome. There are certain regions in our chromosomes known as imprinting control regions, which usually carries a germ. Germ line derived imprinter. The imprinting is mostly mediated by the methylation marker. These imprinting control region are composed of numerous CPG island, which are sites of methylations. Thus, the imprinting control regions are often called as a differentially methylated region. Cytosine and 5 on methylation forms 5 methyl cytosine. The enzyme that catalyzes this reaction is known as a DNA methyltransferase. Yes, adenosyl methionine SAM acts as a cofactor for methyltransferase. Uh, pene- Penetrance and expressivity. The penetrance is the percentage of individuals that shows a phenotype among those capable of showing it. When individuals do not show a trait even though they have the appropriate genotype, when the individual do not shows a trait even though they have the appropriate genotype, the trait is said to exhibit incomplete penetrance. And examples of incomplete penetrance in human is a polydactyl, the presence of extra fingering and toes. The term expressivity is used if a trait is not expressed uniformly among the individuals that shows it. The dominance lobi I mutation in chromosomes is an example of expressivity. This phenomenon associated with this mutation is extremely variable. Some heterozygous flies have tiny compound eyes, whereas others have large lobulated eyes. Between these extremes, there is a full range of phenotypes. The lobi mutation is therefore said to have a variable expressivity. The incomplete penetrance and variable expressivity indicates that the pathway between the genotype and the phenotype is the subject to considerable modulations. Some of this modulation is due to environmental factors, but some is also due to the factor in the genetic background. The clear-cut evidence for such factor comes from the breeding experiment by showing that the two or more genes can affect a particular trait. Genocopy. Genocopy. The phenocopy is a term coined by the Richard Goldsmith in 1935, which refers to the condition where the phenotype of an individual is altered because of an environmental factors, and thus the individual phenotype matches a phenotype which is determined by the genetic factors, though in fact it does not. It is not a type of mutations, as it is non-hereditary. In case of human, the phenocopies are often brought about by the drugs or chemical substances. For example, the corticoids induced diabetes has a phenocopy of genetically determined diabetes. The phocomelia is a rare genetic disorder which the hands or feet are attached close to the trunk, the limbs being grossly underdeveloped or absent. This can also be caused using a drug known as a thalidomide. In case of E. coli, the F plus cells having the fertile factor is converted into the cell which are good recipient and poor donors by a long incubation in the stationary phase. That is, they are F phenocopy. Sex linkage. One day in 1910, the American geneticist Thomas Hunt Morgan peered through a hand lens at a male fruit fly, and he noticed it did not look right. Instead of having the normally bright-eyed eye of the wild-type Drosophila melanogaster, this fly had a white eyes. Morgan was particularly interested in how traits are inherited and distributed in developing organism. He wondered what caused this fly eye to deviate from the normal. Morgan did the same simple breeding analysis to find out more about the white eyes and found the following surprises result. Morgan found this the type of inheritance in different forms of normal expectations of Mendelian inheritance. This is known as a sex linkage. The sex-linked recessive inheritance is also called crisscross inheritance, as X-linked characters are transmitted from the father to the grandson through his daughter. The crisscross inheritance is observed in case of inheritance of color-blind genes in humans. This can be illustrated by the above crosses. Linkage and crossing over. Certain genes located on the same chromosomes tends to remain linked together while passing from one generation to another. This tendency of such genes to stick together in the way in which they enter the cross have been termed as a linkage, and such genes are called as linked genes. In genetic, the complete linkage is defined as the set or state in which two loci are so close together that alleles of those loci are virtually never separated by the crossing over. In case of male Drosophila, this phenomenon can be explained by the test cross as follows. This shows that the gene for great body and a long wing are linked together and do not abide with the Mendel's principles of independent assortment. In 1918, Morgan found a little amount of crossing in the case of female Drosophila, and thus the genes are not completely linked. It is termed as an incomplete linkage. Although it shows a little amount of crossing over, it is not good enough to produce Mendel's hybrid test cross ratio 1 is to 1 is to 1 is to 1. Thus, the law of independent assortment is not enough to describe the phenomenon of incomplete linkage. This phenomenon can be illustrated by the following test cross. Relationship between the linkage and crossing over. Linkage is an exception in Mendel's law of independent assortment, while independent assortment is due to the phenomena of crossing over. Thus, linkage and crossing over are inversely correlated. If the distance between the two genes on a chromosome is longer, it reduces the linkage while increases the crossing over. New combinations of gametes are produced by crossing over, but linkages tends to keep the parent combinations. Sex linked and sex influenced trait. The type of trait which are expressed only in one sex, either in male or female, is known as a limited trait. These are mostly autosomal trait that is not influenced by sex chromosomes. For example, the mutant alleles of breast cancer susceptibility gene BRCA1 is dominantly caused breast cancer in only females but not in males. Thus, this is sex linked. In contrast to this, the second breast cancer susceptibility gene BRCA2 cause breast cancer in both males and females, and thus it is not sex linked. The sex influence trait, on the other hand, can be observed in both sexes, but it is more influenced, but it's more frequently found in sex. This is often termed as a hormone influenced trait. Genotype capital B, capital D, female baldness and male baldness. Capital B, small b, normal hair in male baldness. Small b, normal hair in female and normal hair in male. For example, the baldness is a sex influenced trait which is more commonly in male but can be found in female. If the alleles of baldness are present in homozygous conditions, capital B, the heterozygous female capital B small b may be developed baldness if they have high level of androgen.